So I guess I bring a little bit of diversity to this program. The three previous speakers are from Germany. I'm not from Germany. I'm from the U.S. I would note that Germany is about 1/4 the size of the population of U.S., and there's only 1/4 of speakers from the U.S. here. So there's a bit of a sampling bias, Rajiv. Yeah. So just briefly, I picked up this book right before this session, and the coffee table book that Rajiv was promoting. I opened it up to page 28, and I see Rajiv in a lab coat. Did that really happen? When was he in a lab coat? So afterwards, I'm going to get him to autograph that picture for me. So I'm going to talk about some work that we've been doing in groundnut, and this is really a team effort. And so I just want to up front talk about the major players. Most of what I'm going to talk about is work from David and Soraya Bertioli and Dongying Gao, who's a postdoc in the lab. There's been a lot of other contributors, and the names are up here, and those represent large groups. So each one of those names represents a group of people. And the fundings come from a variety of places. And I'm going to talk about, this sort of interleaves several of previous talks from today and yesterday about accessing wild germplasm and also addressing issues of production, and specifically in peanut, what we're doing to use germplasm collections to address issues in production. So everyone has showed a bottleneck picture, so I figured I must show one as well. So you get the idea that you have some staining variation in an ancestral population. You go through bottleneck, which can be domestication. A bottleneck can also be breeding. Each time you make a cross and make selections, you're imposing a bottleneck. And your surviving population represents a subset of that variation that's in the ancestral population. So, of course, it's a positive thing, right? There's a lot of deleterious alleles that are left behind that you don't want in a domesticated crop, things that shatter and uneven flowering and response to light and things like that. But there are potentially useful traits that we've left behind as well, and that's what we're going to talk about here in the case of groundnut. So just sort of an outline I'm going to talk about. I've already talked about bottlenecks. I'm going to talk about sort of a history of the genus Arachis, which is the genus that contains peanut. I'm going to tell you that peanut is a tetraploid and what we're doing to resynthesize that tetraploid and how we're using genomics to improve peanut. So really, the legumes have become a very rich set of species to do genomics. And a lot of this has been driven by work coming out of the Center for Excellence in Genomics here at ICRISAT. But since 2010, there's been a number of reference genome sequences that have come out, ranging from soybean to pigeon pea, which was a CEG genome. Chickpea was from the CEG. Cowpea was from the CEG. Groundnut was participated in by the CEG International Consortium. And they're all part of a subset of the legumes that we call the papilionoids. And this is where the crop legumes come from, are the papilionoid subset of legumes. And here's the Arachis is out here. It's sort of an outlier compared to some of these other ones that we've worked on. So it's the third largest, missing a T, third largest family of flowering plants. Most of the sequence genomes have been relatively small ones and things that are not too polyploid. That's starting to change, and I'll show you some work that has been going on there recently. So Arachis hypogea peanut is native to South America. It was domesticated in the Andes, probably in Peru. I think everyone knows it's an important protein and oil source, an important subsistence crop in many parts of the developing parts of the world, including Africa. It's a relatively large genome. It's about 2.7 gigabase pairs, so slightly larger than maize. And again, it's an allotetraploid. Genetically, it's been thought to behave in a disomic fashion. I'm going to come back and touch on this a bit later in the talk. But we can refer to it as a disomic tetraploid. And we have hundreds of existing wild relatives and land races in various collections here at ICRISAT in the U.S. and in South America. So it's endemic to South America. Domestication took place over here in the Andes. But the spread of the species, the cultivated species in South America, but also the distribution of the species sort of follows this, where the dark green is on the map. And you can go, this is from Jose Valls in Brazil, out collecting plants. You can walk through fields like this, and you can find wild diploid Arachis species out there. So what do we know about Arachis hypogea? So if you look at the karyotype, so it has two sets of chromosomes, 10 chromosomes each. If you look at the karyotypes of them, 10 of them have large heterochromatic bands, and this is work from Guillermo Sejo in Argentina. And the other 10 have no large heterochromatic bands around the centromeres, or pericentromeric heterochromatin bands. And if you look at the karyotypes of some of the diploids, you can see similarities. So a set of diploids with only 10 chromosomes that have been, we call A genome species. Arachis durinensis has a karyotype that's very similar to a set of the 10 chromosomes in the tetraploid. And from a B set of diploids, you see that there's also similarity in the karyotypes from these species to those in the tetraploid. So just based on karyotypic work going back years, we had sort of an idea of what the progenitors, the diploid progenitors of tetraploid peanut were. And 10 from these A and B diploid progenitors. More recent molecular work from David Bertolli and others, and this is actually looking at intron work, but now been updated with sequencing. What we can do is you can take sequence data, or you can take intron data, band size data, and classify these different diploid species and the tetraploid. So here's Arachis hypogea, one copy. Here's another copy of the Arachis hypogea. These are the two subgenomes. And what you do is have a cluster of B alleles from the tetraploid and B subgenome and B genome diploids, and the A subgenome from the tetraploid and A related individuals. And so this really gave us an idea of what the relatives were, what was most closely related to what the most closely related progenitors of these two subgenomes in the tetraploid were. And this became very useful as we moved forward to begin sequencing the genome. So you can see for the A genome that we have Arachis durinensis, which is a very closely related diploid, A genome diploid, and for the B genomes is Arachis impiensis, which is the most closely related B genome diploid. I should note that the divergence time between these A and B genomes is about 2 to 3 million years. So they're actually very closely related, these subgenomes. And this was complicated when we began looking at doing sequencing the genome of the tetraploid. If you did a standard Illumina approach-based Illumina-based sequencing approach 5 or 6 years ago, when we started this, everything sort of collapsed together, and it was a problem to sequence the tetraploid. So it's really a story of two genomes. You have the A genome diploid, and you have the B genome diploid came together, and there's probably a spontaneous duplication or sexual polyploidization that led to a sexually fertile AABB genome allotetraploid, having representative chromosomes from these two diploids, and about 2.7 gigabase pairs. So work on SSRs and people that really understand the biology of peanut, you have this founding population, you have the bottlenecking event. I think in the case of peanut, this is even a more severe bottleneck that we think about in many other bottlenecks in domesticated crops, because the bottleneck event is also coincident with the polyploid event. And so people that have looked at the molecular evidence for the polyploid event and subsequent diversification within the tetraploid species, and also those that understand sort of the biogeography of these species in South America, have hypothesized that this polyploid event may have been a single event. Of course, there's not strong evidence for that. It could have been a swarm and only a few survived. But regardless, this bottleneck event that occurred in the formation of peanut is a very severe bottleneck. And that means we don't have a lot of standing variation within cultivated peanut to find resistance for pests and the pathogens and the various abiotic stresses that we have that beset production. So the founding population is also sexually isolated from this because of the triploid block, this 4x2x block between the diploid and the tetraploid cultivated peanut. So polyploid domestication were coincident about 9,000 years ago. This is based on molecular dating of the two subgenomes when we published the two diploid genomes a couple of years ago. And earliest radiocarbon dating of what looks to be appears to be cultivated peanuts placed as about the same time, 7 to 10,000 years ago. Okay. So both of the A and B genomes were sequenced separately as a proxy to understand the biology of the genome of the tetraploid, because at the time that was the best way to get to the tetraploid genome, because the sequencing of the tetraploid using the Illumina-based approach just had a lot of collapse between the A and B subgenomes. So we sequenced the A and B genomes independently, and that really gave us a good insight into the tetraploid genome. So these diploid wild relatives that are sexually blocked or isolated from cultivated peanut are a rich source of resistances, resistance to pathogens and pests. So this is just one species. This is Arachis stenosperma. And here we're showing rust, root knot nematode, fall armyworm, and late leaf spot, which is a major problem in some areas of production of peanut. And you can see there's almost what appears, I'm not a pathologist, so if I use the word near immunity, please forgive me for using it incorrectly, but as a person that looks at the genetics as not a pathologist, you have very strong resistance in these diploids to these pathogens that affect the cultivated peanut. And so that's been one of the foci in the lab is using various strategies to access the genes and the alleles in these diploids and bring them in to solve these problems in the tetraploid cultivated peanut. So as I mentioned before, we have very good and accessible collections that have been maintained for decades, curated, and information has been layered on these over time. So the USDA collection was actually located about an hour and a half from our campus in Georgia. It has about 10,000 accessions of peanut and wild relatives. ICRISAT has a very rich set of accessions as well. And in South America and Brazil and Argentina, they also have sets of collections. So there's some work going on in genotyping these collections. So work with Rajiv and Richard Mitchelmore at UC Davis used exon capture of some mini-cores to begin to explore the genetic variation, sequence variation. There's been some genotyping by sequencing of mini-cores. And I think the group here at ICRISAT and others are in the process of beginning to resequence some of the mini-core collections or the entire collections. We know that, so over the past 20, 30 years, there has been some limited use of these wild relatives. It's been very laborious. I'm going to tell you the way that we're doing it currently, but it took several decades to get these genes, in one case for root knot nematode, from a species called Arachis cardiacei into the cultivated peanut. It took several decades to get that there. But it's been estimated that annually this has a 200 million impact for just the U.S. peanut production. And you can imagine what that might be in developing parts of the world if you get these resistances, plant-based resistances to the pests and pathogens into the cultivars that the subsistence growers are growing. So I want to talk about what we're doing to get at that diploid gene pool. So what we're doing is we're basically resynthesizing domesticated peanut. And I told you we have these set of A genome species, we have the set of B genome species, and we're going through is in a somewhat random and a somewhat informed way, resynthesizing AB tetraploids from these different species, choosing diploids that have specific resistances to pests or pathogens, and just making random crosses looking for interesting traits that fall out of these. It's very difficult to look at agronomic traits in these wild relatives because they don't behave in an agronomic cultivated way. It's somewhat easier to look at disease and pest resistances, but even sometimes those break down when you bring them into the tetraploid background. So what we do is make B by A crosses. We use the B genome as a female and the A as the pollen donor, and this is done in a greenhouse. This is a very laborious process. You get an F1 hybrid, which is sterile. Looks very much like the parent, so it's actually we have to use molecular markers to test it. You make cuttings from these F1 hybrids that they don't reproduce by seed, but we make cuttings. We can regenerate from the cuttings. You treat those with colchicine to induce doubling of the chromosomes. We plant those out, get those to induce rooting and flowering. Hopefully there are true tetraploids, and then you can look for fertility through pod emergence or peg emergence, then pods that go into the seed, under the ground, and then eventually you get pod development. Every one of these it's another bottleneck. If you start with a certain number, you lose a number each step of the way. So it's a lot of work to make these B by A diploid crosses, get the hybrid, double it, get the plants to root and flower, to pollinate, and then to get the seeds, and then the entire way to make sure that you're actually dealing with a true interspecific hybrid. So these sterile F1s are very similar to the parents. So this is a stenosperma times valida cross. So here's the two diploid progenitors. You look in the middle and see that the F1 hybrid looks very similar to the parents. And even in flower structure, it's very similar. There's a little difference in flower color. So we use molecular markers. This is sort of an old technology. We now use SNP-based markers, Caspar markers, and so forth to assess the hybridity of our F1s before we start moving them through colchicine treatment and so forth. So as I mentioned, this is Valida times stenosperma. You can look in the F1, you look at the pollen. So here we're studying the pseudocarmine. You see fully formed pollen in both diploids. You look in the F1 hybrid. And this is because the chromosomes don't pair. So even though there's only been 2 to 3 million years of evolution between these, or distance between these two diploids, the chromosomes don't pair in the diploid effectively, and you lose fertility. If you double it, then you regain fertility. So here's this F1 doubled. So it's a velutus and sperma, synthetic allotetraploid. And then that is immediately crossable to the cultivated peanut. So within about 12 to 18 months, we can have tetraploids that are immediately crossable with cultivated peanut. So within these synthetic tetraploids, we immediately see variation for various traits. So here's one of the parents, and you can look down this row in the greenhouse, and you can see architectural variation. So there's some more erect plants that grow in the main stem grows in a very upright way, where others are more lateral spreading. You can see variation in flower colors, maybe due to transposons being released in the hybrid or in the tetraploid, but also you get some variation in flower morphology. Following up on the structural or growth habit variation, you can see that there's variation within sort of the architecture of the stems and the lateral branches coming up from the main stems, the very sort of lateral branching to the more erect type of branching. And all this is immediately visible in those immediate allotetraploids, synthesized allotetraploids. So going from that synthetic allotetraploid back to domesticated peanut is actually pretty easy. I think we often think of domestication as this long, drawn-out process where there's been hundreds or thousands of years of selection by humans on these, going from something that's very wild and weedy and shattering and not photoperiod responsive and all these other traits that you look at as far as domestication to what we have today. In the case of peanut, so here's what domesticated peanut looks like. The immediate synthetic allotetraploid looks something like this. So you have multiple seeds, but they're separated by this long branch between them, so that which is bad for harvesting, right? So if they're separated, and these are underground, they often get separated, so you lose some of your yield. So you want them close together in the same shell. But we can recover this with just a few cycles of selection back crossing to the domesticated peanut. So within just 2 to 3 cycles of selection, you can almost fully recover what a domesticated peanut looks like. So we can do 12 to 18 months to generate these synthetic allotetraploids. A year or two of selection crosses back to domesticated peanut and selection and recover what it looks like essentially as a cultivated peanut, and along the way selecting for disease resistance traits. So it's a very exciting, very rich area to move forward in. So here's some of this is just a subset of the crosses that we've made. So Epiensis and Duranensis, we think those are the progenitors. We're almost certain that those are the progenitors of the domesticated peanut that we grow today. And so we've been using various successions of those, Duranensis and Epiensis, to resynthesize that. And then these are various A-B combinations that we've been using. And some of these we know that there are traits in. So Stenosperma, which I showed you before, has nematode resistance. It has late leaf spot resistance and various other traits. So we cross them to elite cultivars. These is done in the greenhouse the first couple generations, and we move and collaborate with breeders down in Tifton, Georgia, which is closer to Florida, to begin to assess these things in the field and growing conditions. So And what you see is 4 copies of A and 0 copies of B. Between these, so this one's lost all of the versus the rest of the genome. So some of them are recurrent, some of them we find independently. The second part of let me get to the second part of the question by saying we do find some traits appear to map at these regions. And so we don't know if there was selection to become tetrasomic that actually increased the value of a trait or added to the trait. That's something we're interested in looking at. And also, that would allow us to get at the question of heterosis in those. In the synthetic allotetraploids, we also find these things happening within the first few generations. So it happens very rapidly. And we're seeing some association with some of the traits, visible traits in the plants.
Right. Let's give our speaker another applause. Well, yours.